All right, once again, we're going to talk about biotechnology, but this time after we review, we'll talk about safety and ethics of biotechnology, an area where there's uh, less to see maybe than some people think. Uh, we'll describe how science helps us evaluate ideas, basic kind of how science works, and then describe efforts that have been made to ensure the safety of uh, biotechnology, genetic engineering, and things like that. So we've discussed a lot of techniques, and some of them are widely used and others are less widely used. Some have been wildly successful, others maybe less. I've talked about cloning an organism where we just replace the nucleus of an egg cell with a nucleus from a body cell and then implant it in a surrogate host. I've talked about creating recombinant DNA and recombinant cells and how that's used to do things like make insulin for diabetics, to put genes in plants and animals, that this has been very, very successful and is widely used. Talked about gene therapy, where we use genetic engineering to replace defective genes and with functioning copies in patients. This has been much more difficult to do than people originally envisioned, but there's been some success, but much less than originally thought. Polymerase chain reaction, we use primers to grab DNA, and then an enzyme copies DNA to produce unlimited amounts. Similar technique using DNA polymerase and nucleotides, we can sequence DNA doing chain terminators so that we separate fragments that end with a fluorescent dye of a particular color. We separate them by size, and the computer reads the sequence. Easy to do, very successful, and very helpful. I've talked about DNA profiling, using several techniques to identify DNA of individuals. Um, I don't want to say wildly successful, but very successful, and that it's helped us identify cases of mistaken convictions and help us really identify individuals from a biological sample at all. So, and if you think of safety and ethics here, this has really been an area where DNA has really improved the quality of the justice system and its ability to identify individuals that have caused crimes, particularly crimes involving biological samples. And so blood, semen, those types of things. Really, in this case, making our justice system safer and more equitable. And so, if nothing else, we can identify an area where biotechnology has benefited all people by making our justice system better. But there are lots of concerns about the safety of genetically engineered products and foods, particularly recombinant DNA or genetically modified organisms or transgenic organisms, particularly about the ability to um, do DNA testing to identify individuals for uh, things they don't want to be identified for. Um, and so there's been a lot of fears about genetically engineered foods. Is it going to change people that eat the food? Is it somehow much more dangerous than foods that are produced more traditional ways, what are often considered natural? Are we going to create an environment where one or more genetically engineered organisms takes over, essentially um, out competing all existing organisms? So we can think of lots of different fears. There's always been fear of technology, fear of change, and that new technologies might bring with them new dangers. Um, but we have a really good tool for identifying these things and making decisions. This is what science is designed to do. It's to help us choose the best alternatives based on evidence. Um, Ideas about harm, these are ideas that are possible, but we can weigh how likely they are, and how different these new organisms are. And so, we want to identify the most likely outcomes and potential consequences. And in science, we use experiments to test ideas. We call those ideas hypotheses, and we use the evidence from experiments or the other available evidence to identify which hypotheses are most likely wrong and which are most likely right. And in science, that stuff is available. It's available for other scientists to look at and to review. And so this is really important to recognize that science is based on evidence, and that evidence is open to other scientists. Um, Sometimes the evidence can be misconstrued, but it's really important to look at that evidence. And from the beginning, um, there scientists identified certain possibilities that might happen, and they've um, right from the beginning of genetic engineering, there were conferences to among scientists to kind of set out a regulatory pathway and um, to make sure that we were doing our best to check so that safety could be ensured. And so um, here I have a picture from a conference on regulating biotechnology. There were in the early 70s, before this all got going, there were big conferences where they dealt with these questions, tried to identify possible outcomes, and then tried to identify the likelihood of those occurring. Um, with safety issues, we have in place methods for testing genetically engineered organisms. Those organisms require extra approval. They are not automatically approved. They are not innocent till proven guilty. This is one area where we do our best to ensure things. Um, there are ethical issues, I think, that are more important that are associated with genetic engineering, like who might be injured, who might be affected by this, who owns DNA, who benefits, and then who decides. There are some examples that are really important. Um, there's a link here if you watch the PowerPoint. If you go to the PowerPoint and put this in the play mode and hit on devious defecator, it'll talk about a use of DNA testing that was ruled illegal. And so it was to identify someone for uh, a minor annoyance. They required all employees to have their DNA sampled and tested and compared to a biological sample that was left in a warehouse. That was determined to be illegal. The forced testing of employees was not something that should have been done. Likewise, another kind of really thorny ethical issue is who owns DNA. Most often, scientists just find a gene that's useful. They don't invent it. They are able to patent some of these things. And so sometimes this leads to someone making unavailable something that could easily be available and could do a lot of good. Best example of this I can think of is uh, the BRCA1 gene. This is a gene, a tumor suppressor gene, a mutation in a tumor suppressor gene that's linked to breast cancer. And so the gene, the mutation has been patented. There's a company that owns the patent and uh, requires a hefty fee to test women for this gene. Now, they didn't invent the gene, but by patenting it, they control the rights to test for it. That really restricts the amount of tests that are done. It also, um, the cost is really high. Um, and so uh, those are pretty serious kind of limitations that affect everybody. And the people that own the gene or own the patent on the gene didn't really invent it. Um, but they're making a lot of money on it. They were just the first one to find it. So we're regularly going to see new DNA technologies. It's crucial that we look at those and see who can benefit from them and what kind of regulatory stance we need to take on them. A good example of this is the CRISPR technology. CRISPR stands for clustered regularly interspaced short palindromic repeats. These are enzymes found in bacteria and archaea that are used to destroy invading viruses. The people that came up with this didn't invent it, but they identified the enzymes and got them to work in a test tube. And what these enzymes do is they open DNA and they can edit genes. They can cut pieces out, they can put new pieces in. Really powerful technology that can be used to shape organisms. Potentially great benefit, potential misuse. We could do this on an embryo to, put in features that we liked. Um, it's your book talks about it a little bit. I didn't put it in for the main sequences because it's not widely used sequence of DNA technologies that we're going to be tested on because it's not widely used yet, but it is up and coming and will be really more and more important over time. Um, but sometimes the fear of these are really overblown. And so a good example of this is the COVID-19 vaccine. These are messenger RNA vaccines that what's injected to a person are antigens, or really are messenger RNA molecules that will cause cells to produce proteins that are antigens for the virus. These will be easily recognized by immune cells, and these vaccines are easier and faster to make and produce really good responses. Okay, they are not live vaccines, so you can't get a COVID infection from this. It's just a messenger RNA for part of the virus for the surface molecules. They've been proven very safe and effective. And here I have a table of the data here. Let me go through this. This is from COVID 19 during the period of January 2nd to July 2nd of 2021. So this was right after the vaccine was introduced when COVID was strongest and infecting the most people. And it shows um, in the final column are the percentage of COVID-19 deaths as a percentage of all deaths. And this was done in Great Britain. And so for the time period they're talking about, there were 214,000 deaths in Great Britain in that 6 month time period. Almost 20% of them, 19.3%, were due to COVID. So many, many COVID deaths, 50,000, uh, almost 20%. If you looked at just unvaccinated people, people that didn't get the vaccine during that time period, there were 65,000 total deaths and 36,000, almost 39,000 of them were due to COVID. That's 37%. Okay, more, almost double the number of deaths due to the percentage number of deaths due to COVID. So unvaccinated people really suffered from this. If you looked at people that had their first dose but died within 21 days of that first dose, that was 23,000 or 15,000 people and 23%. So slightly less than the unvaccinated people. So even the vaccine, even within the first 21 days, provided some protection compared to none. If you looked at people after 21 days of their first dose, um, the number of deaths was only 7,000, and that was only 10%, less than 10% of the deaths in that cohort. So the vaccine, the first dose, provided very good protection, almost double the protection after just 21 days. Um, for people that had two doses, only 1.6% of them died of COVID in the first 21 days, and after 21 days, it was less than 1%. Two COVID vaccines really protected you. That's really, really strong evidence of a protective effect. And that's overall deaths. So really, really powerful, powerful evidence that the COVID vaccine was safe and effective. I'll stop there. We'll talk about this maybe a little bit more when we review for the exam.